Decoding the molecular ballet, the intricate process of protein synthesis. 1. Introduction. Biological choreography. Protein synthesis is a meticulously orchestrated cellular dance, a process through which the instructions encoded in DNA are translated into functional proteins. This intricate molecular ballet occurs within the cellular machinery, involving multiple steps and key players. 2. DNA to RNA. Transcription. Initiation. The process commences with transcription, where a specific segment of DNA unwinds. An enzyme called RNA polymerase recognizes and binds to the promoter region, initiating the synthesis of a complementary RNA strand known as messenger RNA, mRNA. Elongation. As RNA polymerase progresses along the DNA template, it synthesizes the mRNA strand by adding complementary RNA nucleotides. This elongation continues until it reaches the termination signal. Termination. Transcription concludes as RNA polymerase encounters a termination signal on the DNA. This signals the release of the newly synthesized mRNA strand. 3. mRNA and the ribosome. Translation. Initiation of translation. The mRNA, now carrying the genetic code, travels to the ribosome, the cellular machinery responsible for protein synthesis. Translation begins with the binding of the small ribosomal subunit to the mRNA, followed by the attachment of the initiator tRNA, transfer RNA, carrying the amino acid methionine. Elongation. A larger ribosomal subunit joins the complex, and the ribosome moves along the mRNA in a process known as elongation. As each mRNA codon is read, a corresponding tRNA delivers the specific amino acid dictated by the codon. The ribosome catalyzes the formation of peptide bonds between successive amino acids, creating a growing polypeptide chain. Termination. Translation continues until a stop codon is reached on the mRNA. Release factors recognize the stop codon, leading to the termination of translation. The newly synthesized polypeptide is released from the ribosome. 4. Post-translational modifications. Folding and processing. The freshly synthesized polypeptide undergoes post-translational modifications. This includes folding into its three-dimensional structure, guided by chaperone proteins, and, in some cases, the addition of functional groups or removal of segments. Golgi apparatus and secretion. In eukaryotic cells, proteins may pass through the Golgi apparatus, where further modifications, such as glycosylation, occur. Fully processed proteins are then packaged into vesicles for secretion or incorporated into cellular structures. 5. Importance of protein synthesis. Functional diversity. Proteins play diverse roles in cells, serving as structural components, enzymes catalyzing biochemical reactions, signaling molecules, and carriers of genetic information. Protein synthesis is essential for maintaining cellular functions, growth, and adaptation. Cellular machinery. The machinery involved in protein synthesis, including ribosomes, tRNA, and enzymes, is integral to cellular activities. Mutations or disruptions in this process can have profound effects on cellular function and may lead to diseases. 6. Regulation of protein synthesis. Gene expression control. Cells exert tight control over protein synthesis to adapt to changing conditions. Gene expression is regulated at multiple levels, including transcriptional control, initiating or inhibiting transcription, and translational control, regulating the efficiency of translation. Signaling pathways. External signals, such as hormones or environmental cues, can trigger signaling pathways that influence protein synthesis. This allows cells to respond dynamically to their surroundings. 7. Errors and quality control. Proofreading mechanisms. Both transcription and translation undergo proofreading mechanisms to ensure accuracy. Enzymes, such as DNA polymerases and aminoacyl tRNA synthetases, monitor the fidelity of nucleotide or amino acid incorporation. Ubiquitin proteasome system. Misfolded or damaged proteins are targeted for degradation by the ubiquitin proteasome system. This quality control mechanism helps maintain cellular integrity. 8. Conclusion. A molecular symphony. Protein synthesis is the molecular symphony that orchestrates life's processes. From the transcription of DNA to the folding and processing of proteins, this intricate dance ensures the continuity and functionality of cellular life. Understanding the nuances of protein synthesis not only unravels the mysteries of cellular biology but also holds the key to advancements in medicine, biotechnology, and our broader understanding of life itself.